So there's two slides here. Yeah, so there's two different slides you're working on there. All right, so just send in that number there on your clickers. So the number is which step is where the blood is coming into the heart from the lungs. All right, anybody else? I think that's about everybody. Oh, there we go. 60. Okay. All right, well, let's come back together, guys. So we're a little bit all over the place there. Maybe about 30% of you saying 5, some saying 6, some saying 7. We'll get there in a sec. Yeah. So yeah, let's look at that real quick, and then we'll kind of do the normal intro stuff. But I am feeling awkward having my mask down because it's been 2 years. So just to, but yeah, if some of you weren't here last time, I polled the class to see how you guys felt about that with the new guidelines, and yeah, over 90% of you said either you don't care or take your mask down. So I'm trying it out, but it does feel weird now. That's been so long, but all right. Anyways, here we go. Okay, so yeah, so just walking through the cardiac cycle here, we can see the blood out in the lungs here, obviously, and then yeah, well, sorry, not out in the lungs. That is out in the systemic circulation, right? Yeah, so this is blood in the systemic circulation. So this would be like out in the body, right? And then it's coming back via the vena cava, right, into the right side of the heart. So we got the right atrium there, and then it's going to go into the right ventricle in step 3. Right, that right ventricle is going to pump. Well, first of all, it had to go through this valve. What's that one called? Tricuspid, okay, to get to the right ventricle. Then it's got to pump out, go through this valve here. What's that one called? Pulmonary valve, okay. Now it's going to go out to the lungs. So we see that. So that deoxygenated blood leaving via the pulmonary trunk and the pulmonary arteries. It's going to go get oxygen in it and then come back here. Now it's red because it's got the oxygen in it, okay. Then it's going to come back via those pulmonary veins, which are unique because they're veins that carry oxygenated blood, right? And then we see the blood coming back into the left atrium right there, right, from those pulmonary veins, okay? So that's where I would say the answer to the clicker question would be number 6. It's still happening in number 7. I mean, that's this whole thing is all these things are happening at once, right? That's kind of how all of this works. It's not like step by step. It's always all happening, right? There's blood moving the whole time. And then, yeah, and then we fill up the, yeah, so then we fill up the left ventricle fully here, which this is just kind of a weird view because it makes the left ventricle look smaller than the right ventricle for some reason. I don't know why this image does it that way, but it's definitely not the way that it is. And then, yeah, and then, well, first of all, we had to get through this valve. What was that valve called there? mitral valve, okay, or the bicuspid, and then finally the ventricle contracts, pumps through that valve. What's that one called? Aortic. Good. Now we're sending blood out the aorta, out to the systemic circulation. Okay. Good. Questions on any of that? Go ahead. What are steps 4 and 5? Steps 4. Step 4 would be. Oh yeah, because there's actually only 4 images, but there's 5 steps. Huh? Yeah. Don't get too hung up on the numbers as long as you know like what's going on. Is there a picture you're wondering about what's happening? Yeah. As long as you understand where the blood's going in each step, you're fine. Yeah. I'm not sure why there's 4, why there's 5. This is right from your book though, if you want to pull up that figure and see exactly what it says. But yeah. Yeah. Other questions on that? Go ahead, Mallory. Yes, comes back into the left atrium, left ventricle, and then out to the body. Yeah, good. Okay. All right. Yeah, and then we'll just finish up with this here before we move on to the notes that we're supposed to be for today. So when we look at the heart, it's really, and the whole vascular system, it's really 2 separate circuits, right? So we have the pulmonary side and the systemic side, right? So the pulmonary side is from the heart to the lungs and back to the heart, right? This side is very low pressure because it doesn't have far to go, right? The lungs are wrapped around the heart, right? It's right there, okay? Don't have far to move that blood, so that's a much weaker side. The right ventricle is much weaker than the left ventricle, okay? Out the pulmonary trunk, pulmonary arteries, to the lung capillaries, back via those pulmonary veins, and then back to the left atrium, okay? Systemic side is obviously the heart creating our blood pressure, sending it out to the rest of the body. Okay, this is much higher pressure, much farther to go, much more gravity to deal with. If you think about it, it's got to get all the way down to our toes, but then back up, right? So we've got a lot of gravity issues to deal with there. And also just right from the heart up to our head, we got to fight against gravity to get it there as well. So we need all that pressure, okay? And so then we go aorta, down to arteries, down to capillaries, veins, vena cava, as well as the coronary sinus, back to the right atrium. Okay. All right. Questions on any of that? Okay. We're going to hop over to the other notes, but while we're doing that, just a reminder of what else we got coming up this week. So the lab quiz tomorrow will be over those coronary blood vessels that I emailed you about. Okay, so there's 7 of them that you need to know from that page 20 on your handout. Okay, so those ones, as well as all of those thoracic arteries that you, thoracic and upper extremity arteries that you learned last time in lab on the cat. Okay, and then again, the ones that say what tissue is supplied by it, then you need to know it. Okay. If it doesn't say anything next to it as far as what it supplies, then you don't need to try to look up what it supplies. You just don't have to know that part. Yeah, Mallory?
You need to know that circulatory system, like the heart.
Yeah, no, actually not. Yeah, not for this one. You will eventually, but not for this quiz. Yeah.
I don't know if you can say, but for the images of the cat arteries, would it be more of the zoomed out view so you can kind of differentiate like where you're at on the cat, or is it like zoomed in?
Yeah, you can definitely get your orientation. You can see what, you know, what side of the body it is, and yeah, you can definitely, you should be able to count back from all, because that's really what it is. And when you get to those arteries in the arm, it's really just about the order of things for the most part, right? You kind of just have to memorize this one comes first and the next, and then so yeah, you can definitely see all that you need to see with that. Yeah, in one image. Yeah. Yeah, very similar to like what's in the textbook. Yeah. Yeah. Go ahead.
So just to confirm for the lab quiz tomorrow, then it's the 7 coronary arteries, and then the cat thoracic arteries, and is that it?
Yes. So all those arteries that you learned last time. So the ones that come off the heart there, and then those big branches, and then it goes up into the arm, as well as I think there's a couple in the neck too, right? Where it splits off to the, yeah. So all of, yeah. Yeah, not yet, but you will. That's what you'll dissect this week, is the ones below the waist. Yeah, and above it too. Yeah, good. Other questions on that? Yeah, go ahead. Yeah, this is printed, so it's a printed color image. So you kind of get to go at your own pace. And the one next week will be the same way. The 16.1 will be printed as well with a color figure. Yeah. Mhm. Good. Okay, go ahead. That also is for the practical, but and for the quizzes. Yeah. Yeah. Okay. Other questions? All right. Okay. Well, let's dive into this here. So today we're going to get into a little bit more of the physiology of the heart and specifically talking about the electrical properties and how action potentials look in the heart. And so before we do that, we're going to review a little bit about action potentials that we learned last semester. So that would be action potentials that we'd find on a skeletal muscle cell or on a neuron. Okay. So you guys remember these images before. This is the same slide I had from last semester. So really, we got these different states of the action potential, right? We got resting state. Number 2 is depolarization. 3 is our repolarization. 4 is our hyperpolarization. Okay, so we got those different phases, right? And then the whole question of what causes all of this. Well, what causes all of this is, first of all, permeability changes in the membrane, and that's permeability to certain ions, right? Because we know that ion flow is what leads to all those little changes in membrane potential with depolarization, repolarization, and hyperpolarization. So in order to make ions flow, we got to change the permeability of the membrane to allow them to flow. Okay, so what's going to dictate the permeability is a bunch of different voltage-gated ion channels, the ones we talked about last semester. All right, so we got that sodium channel, right? How many gates does the sodium channel have within it? Some of you might have thought I said, how many states can the sodium channel be in? That would be 3, but it has 2 gates, right? It has the inactivation gate and the activation gate, okay? And then we have the potassium voltage-gated ion channel. And that one's just got the 1 gate, right? It's either open or closed. The other one can be activated or inactivated or in its resting state. Okay, and so you can see how this graph kind of matches up with that one. We can see that we have a big spike in sodium permeability during depolarization, right? And then we have a little bit of a spike in potassium permeability during both repolarization and hyperpolarization, because the flow of those ions are what cause those different phases. Okay. Yeah, so that's just a little bit of a review for that, okay? And then we're going to transition into talking about what the heart action potentials look like, which is a little bit different than this. Okay, questions on any of that before we keep going? Go ahead. Sodium caused the influx of sodium caused the depolarization. Yep, and then it's the efflux of potassium that causes both repolarization and hyperpolarization. Okay, so, and then also it's important to review how muscle cells contract, because we're going to be talking about that again with cardiac muscle cells, okay? So we had this whole thing last time, that whole excitation contraction coupling, as well as cross-bridge cycling, all of that, actin and myosin binding, calcium's involvement, all of that, right? So yeah, we have a very similar setup in the heart muscles, and so we'll get there in a sec. Okay, but one thing that's interesting to note here is the length of an action potential, including the whole refractory period, when we're talking about a muscle cell or a neuron, is only 1 to 2 milliseconds. Okay, that's the whole thing, right? This is from the beginning of the spike of the action potential till the end of hyperpolarization. Okay, so a single twitch of a muscle is about 20 to 100 milliseconds. So that means many, many action potentials occur to lead to a single twitch of a skeletal muscle, okay, to allow that tetanus-like contraction, to get that nice smooth contraction. We get thousands of action potentials causing that, okay? And in the heart, it's a little bit different than that, okay? So in the heart, a few things that are different. Number 1, action potentials start in what we call pacemaker cells, okay? So we have these special pacemaker cells in the SA node and the AV node. You'll see the full written version of those in a couple slides, but it's the sinoatrial node and the atrioventricular node. These have special cells that depolarize on their own, okay? This is like, anybody seen the old Indiana Jones? The guy rips the heart out of it. And it's just, it's like still beating in his hand, like after it's ripped out, or Mortal Kombat, I guess, if anybody's video game people, it's the kind of same thing. There's actually some truth to that, right? Because actually the heart literally contracts on its own. So if we could disconnect it from all the nerves, it can still contract. And it's because of these autorhythmic cells that depolarize on their own. Okay, and so that's where the action potentials are going to start in the heart. So we don't need external input from the heart. And then it's going to pass through those gap junctions we've been talking about for a while, going from cell to cell. Okay, so that action potential can spread. Okay, and then during resting membrane potential, we always have negative on the inside. Okay, same thing as before. And then we have lots of potassium on the inside, lots of sodium on the outside. That's no different from skeletal muscle cells or neurons. That's the same. Okay. When it comes to the depolarization part of a cardiac action potential, still caused by sodium rushing in. So that's exactly the same. Okay. So that's going to be the same as what we just saw with the skeletal muscle and the neuron. Okay. But then in cardiac muscle cells, we have something called a plateau phase. So it spikes up, but then it stays flat for a while. Okay. That's totally different than skeletal muscle. And that's going to be caused by calcium channels opening up and allowing calcium to rush in. So that's really the only major difference here so far in how heart cells do it versus how skeletal muscle cells do it. Okay, and then repolarization is the same as neurons and skeletal muscle cells. It's all caused by potassium flowing out of the cell. Okay. So that next slide is some labeling of that cardiac action potential. So go ahead and label that. And there's a clicker question there on the top that you can answer as well. So go ahead and work on that with your team. And I'll open this up so you can send in a number.
I want to use their copies.
Going good. How are you? Yeah, shoulder okay? You know if you're hurting, a little spasm. Oh yeah, I get that too. Yeah, need to crack your thoracic spine maybe. That's usually what fixes it for me. So you're filling in which ion flow leads to which step with 1, 2, and 3. That's what you're filling in. Don't wait for it to uncover. Write it in there. Which ion flow leads to 1? Which one leads to 2, 3? And then you can answer that question on the top on your clickers. All right, give you about 15 more seconds to send that in on your clickers. All right, got 63 of you. I think there's a few more. Go ahead and make sure you hit the send button with this since it's a number. This might be the most clickers we've had in here for a while, 66. All right, 92% of you are saying 2 there. Okay. All right, good. So what's going on in number 1? What ion flow is leading to that? Sodium going which way? In. Okay. So we got depolarization that's caused by sodium influx. Okay. And then as you guys said, number 2, is calcium influx. Okay, that's our plateau phase. Okay, and then number 3 would be caused by which ion going which direction? Potassium going out. Okay, absolutely. That's repolarization caused first by stopping that calcium influx, but then by allowing potassium to exit. Okay, if you just look at the x-axis on this graph and then go back to the one that showed the skeletal muscle or neuron graph, how long is this one compared to that one? Longer, shorter? The other one was like 2 milliseconds, right? This one's 300. Okay, so it's incredibly longer. It's way, way longer, right? And that's why this looks like a straight line, has no slant to it, because it's the x-axis is so much more expanded. Okay, so yeah, so we're looking at somewhere around 250 milliseconds. Okay, so that's the whole contraction of the heart. Okay, and it's important that this is so much longer because we don't actually want tetanus to happen in the heart. Okay, tetanus is a smooth contraction that occurs from a bunch of different stimuli, all building tension on top of each other, and that would be like a bunch of little muscle contractions, all gathering together to produce force. We don't want that in the heart. We just want one solid pump, right? And that's so this action potential allows for that, right? Because we have this huge refractory period, right? That means as long as we're anywhere in that refractory period, it doesn't matter if we try to send another signal down those heart cells, it's not going to do anything, right? It's not going to depolarize those. It's going to keep those from allowing another action potential to happen. And so we just got that one action potential. The heart can slowly build tension and it can all contract at once. Okay, questions on that so far? And I won't tell you exactly why yet, but it is important that we have this tension graph here on this graph too. Right? And we see that tension is rising during that plateau phase, right? So we'll talk about why that is in a second, but just wanted to point that out for now. Okay. Everybody got this? Okay. All right. So if you put the two graphs on the same x-axis, you can tell there's a big difference, right? So this is what the skeletal muscle action potential looks like if we have it on the same x-axis as this one, whereas this one is way spread out, right? So just a tiny little blip on the left, whereas this long kind of plateau on the right. Okay, so talk about this question with the people on your team. This refers back to that tension building in the heart muscle. Talk about this, decide what you think the answer is, and go ahead and vote.
Sorry, you said what's the process causing polarization, hyperpolarization? Okay, so it's just arteries on the cat, right?
Those are two different questions, right? Yeah.
The cat is just arteries.
I just started, no veins. Yeah. All right, go ahead and send in your answer there if you haven't yet. Need about 20 more of you still. Go ahead and send that in. All right, anybody else? Still about 6 more of you need to send it, looks like. Okay, so 75% of you are saying D, that the tension building is related to calcium influx. Okay, but the next step, I want somebody to raise their hand and tell me, explain that. Why is that? 3/4 of you are saying it's related to that calcium coming in. Why? How does calcium? Go ahead, Ava. The sliding filament theory, it definitely does. Good. Okay. Anybody else want to add a little more detail for us? So 75% of you say that's the answer. Is that just because it's happening at the same time the calcium's inflowing here? Is that why most of you know that? Okay, but what, okay, that's good. That's a good observation for sure. Absolutely. Yeah, nothing wrong with that at all. But the question is, so calcium is keeping the cell depolarized because we're sending in a positively charged ion. So it's staying depolarized. So it's doing that to the electrical membrane of the cell, but it also has a job to do when it gets in there. What does calcium bind to inside of muscle cells? Troponin, okay? And if it binds to troponin, what does that mean can now happen? Yeah, actin and myosin can bind, right? Every new calcium that's bound to a troponin means we can bind one more myosin head to actin, right? And that's what's giving us force in the muscle cell, right? And so as that calcium's flowing in, it's not just doing that for this membrane potential, it's doing it so it can also bind to troponin. And then we got another myosin head contributing to the force production each time calcium binds to troponin, right? So it's important that, and part of this is because we actually, the sarcoplasmic reticulum, which is where we store the calcium inside of our muscle cells of the heart, is not as developed as that of the skeletal muscle. And so it doesn't release as much on its own. So we actually have to rely on that extracellular calcium to come in and help us build force. But we also do want to build it slowly because we're kind of waiting for the rest of the heart to catch up so it can all can contract at once, right? So this is kind of all of that together. So D is definitely the right answer, because calcium's got to go in and bind to troponin, give us more actin myosin binding to give us more force. Okay, questions on that? Go ahead, James. Somewhat, yeah, it is definitely connected as well. Yeah, it's definitely related to it because we don't want to have tetanus. We want to have this like kind of nice long drawn out action potential. So it's connected to the fact that we have a plateau. It's not necessarily connected to the fact that we're getting more tension at the same time, but it is connected to to calcium coming in the plateau, but probably not directly to the tension part. Yeah, good question. Other questions on that? Okay. All right, so when it comes to excitation, contraction, coupling, which is that whole process of preparing for contraction in muscle cells, there's a couple differences between skeletal muscle and cardiac muscle, okay? So remember that action potential is going to come down the neuron, cause acetylcholine release. Acetylcholine is going to cause the action potential on the muscle cell. That action potential is going to dive down those T tubules, cause calcium to be released from the sarcoplasmic reticulum, right? So that's all this part, right? So calcium is released from the sarcoplasmic reticulum, that's the SR, but in cardiac muscle, and it enters, so a couple of things, the sarcoplasmic reticulum enters the sarcomere only once instead of twice. Okay, so just the sarcoplasmic reticulum just doesn't have as much access to where the actin and myosin is. Okay, and like I just said, when we were talking about the last slide, the SR of cardiac cells just doesn't have as much calcium in it. Okay, so 10 to 20% of the calcium that is important for allowing actin myosin binding has to come from the outside of the cell during that calcium influx that we see during the plateau phase. Okay, so we need that extracellular calcium to help us produce force in cardiac muscle. Okay, questions on any of that? All right. We're done, right? Yeah, go ahead, Trevor.
Are we ever going to look at like diagrams of what that looks like?
You mean the
In terms of the sarcoplasmic?
oh yeah, like how it goes in once instead of twice. Yeah, I don't think we have a really good image of that. one of the best images might be on the last labeling slide we did of the previous notes. That kind of showed the SR, but I don't think it really shows entry points into the sarcomere because that's on a more micro level than that image is. But yeah, so I've never really seen a good image of that. Actually, yeah, it's a great question. Yeah.
I guess, like, what does it mean that it enters once inside?
Yeah, I mean, just think about it in terms of like, we don't get as much calcium from the sarcoplasmic reticulum because it just doesn't have as much access to the sarcomeres as it does in skeletal muscle. Yeah, good question. Yeah, other questions on that? Okay. All right, so we'll talk a little bit about the whole conduction pathway now in the heart. Okay, and so here's that SA node again. You can see there it's a sinoatrial node. That's kind of where the rhythm starts and where the electrical activity starts in the heart. Okay, this is in the upper right of the heart, upper right side of the atrium, right by where the vena cava dump in. Okay, the function of this is known as the pacemaker of the heart. Okay, and when it fires, it causes atrial contraction, as well as starting off the action potential of the whole cardiac cycle. Okay, so that's the first part to fire. And then we got the AV node, the atrioventricular node, which is also full of autorhythmic cells, the ones that depolarize on their own. This is right next to the tricuspid valve. Okay, so this is like between the right atrium and the right ventricle. Okay, this is going to receive the stimulus from the atria coming down that started in the SA node. And that's going to send the signal to the ventricles. Interesting thing that happens here is this actually kind of pauses the signal for a very brief period of time, 0.1 seconds, which allows for a little bit of filling time, right? Because we don't really want the atria and the ventricles to contract at the same time, right? We want the atria to kick a little bit, push that last bit of blood in, wait about 1/10 of a second so the ventricles are completely full, and then let the ventricles contract so we can pump the blood out. So the AV node causes that little pause that we need to fill up the heart. Okay, from there, it goes to these structures called the AV bundle. And this is the superior part of the interventricular septum, right? So that septum, that chunk of tissue that's between the left and the right ventricle, this is where the AV bundles are, okay? And then those go down into the bundle branches, which now this splits into two sides, one for the left side of the heart, one for the right side of the heart, still kind of in that spot between the two ventricles. And then finally, we get to what's known as the Purkinje fibers. And the Purkinje fibers wrap around the rest of the heart. So everything so far is just going down the middle part of the heart. This wraps around the rest and causes the rest of those fibers to contract. Okay, these depolarize super fast. Okay. And this makes sense because the whole top part of the heart is already depolarized. It's already starting to contract. So since these ones are the last ones to get the signal, we want this signal to travel rapidly. So the last part of the heart can kind of catch up with the top part of the heart and it can all contract together. Okay, if you're done with this, you can go to the next slide and you can fill that in and answer that question there on your clickers. All right, so check with your team, make sure you're on the same page on the next one as well. So you're filling that in, sending that answer in on your clickers. All right, go ahead and send that in. Go ahead and send your answer to that question there. Which one causes a slight pause in electrical conduction to allow filling time? All right, let's see what you guys said here. Good, yeah, so pretty much everybody, 97% is saying B there, okay? And so first of all, what's A called? SA node, okay, and then B is called? AV node, okay, and then we work our way down to the atrioventricular bundle or just the AV bundle. Okay, and then from there we split off into two sides, one for the right, one for the left. Those are the bundle branches. Okay, and then from there we wrap around the rest of the heart, and those are all called the Purkinje fibers. Also have this other name, subendocardial conducting network. You can call them the Purkinje fibers. And these are those little papillary muscles we talked about last time. Those are connected to this conduction system as well, because those have to contract at the same time that the ventricles contract to do their job. Okay, questions on any of that? All right, so that signal just spreads around like that. And if we were to lay out the action potentials of each of those little sections in a time-aligned fashion, then it looks like this, right? I haven't showed you what pacemaker potentials look like, but this is what the action potential of those AV node and SA node cells look like. It looks a little bit different than what we've seen so far. So we really got three different types of action potentials that we're talking about. One, skeletal muscle and neurons looked like last semester. Then we have the one with the big plateau, which is the cardiac myocytes. And then we have this one, which is the pacemaker cells. Okay, so number one is the SA node. All right, that's going to be our first thing to cause an action potential. We can see that directly leads to number two, which would be which cardiac muscle cell is firing first. And number two, what does the SA node cause? Atrial contraction for sure. So that's the atrial muscle firing. Okay, and then that atrial muscle is going to spread down to the next autorhythmic part that causes a slight pause. What was that one? Yeah, so that's our AV node, right? Oops, node, okay. And then lastly, we finally get the ventricles to contract. That's that big one down there at the bottom. So we get the ventricular contraction there at the bottom. Okay, so you can kind of see how all those different action potentials are working together and kind of one's leading to the next as that signal spreads down. And all of this is really only about 350 milliseconds. Okay. All right, questions on that? Go ahead.
So we come up with a pacemaker, is that because these aren't working correctly, or is that?
When someone has a pacemaker, yeah. A lot of times it is because that SA node isn't working correctly. Yeah, it's common after a heart attack, you get some damage to some of the conducting system of the heart. It could be that the AV node's not working properly. It's actually depolarizing faster than the SA node. And so we kind of have to give it this other feedback to be able to start in the right spot. There's multiple reasons, but something in the conducting system is messed up. And so it's like this. Yeah, and sometimes the conducting system isn't working, or at least the autorhythmic part of the SA node isn't working at all. And so then we got the pacemaker taking the role of that completely. And so, yeah.
My sister got one issue.
Okay, wow. Does it also have a defibrillator in it too? Okay, yeah, so a lot of them, if people have congenital heart conditions, they have the pacemaker part, which does keep the heart going at the right beat. But then also sometimes those same patients are more susceptible to their heart stopping. And so instead of putting the big pads on them, they have a little defibrillator built into that. It'll actually shock them. I don't know if your sister's has that or not, but has she ever been shocked? Do you know?
I don't think
Okay, yeah, so you would know for sure, because it probably would have happened by now. But yeah, some people, I had a friend that had one like this, and when his heart rate would get too high, at sometimes he would actually get shocked. And it is like, it's not as strong as the ones in the AEDs in the box, but it does the same thing. So yeah, interesting. Yeah, Zoe? Yeah, they do. Yeah, and a lot of that just has to do with the size of what we're measuring there, right? So we're measuring many more cells spread out over a larger area in the ventricular muscle versus the atrial muscle, where we just have that little small section. So that's probably the biggest reason for that is what's actually being measured there. But yeah, great question. Yeah. Other questions? Okay. All right, so then let's look at our last type of action potential that you need to know, which would be in these autorhythmic cells. Okay, so these are non-contractile cardiac muscle cells. So they don't contract, they don't have the actin myosin thing going. Their role is to depolarize and start this rhythm of the heart. Okay, so set the pace of the heart contractions. Okay, again, those locations we just talked about, the SA node, the AV node, even some of those AV bundle cells are autorhythmic as well. So the key factor is that the action potential begins on its own. And the interesting part about these is there's really no resting membrane potential. Okay, when we go down between action potentials, it never flattens out in the middle. It just starts coming right back up after it gets to the bottom of the repolarization. Okay, so it repolarizes, goes into hyperpolarization, but that hyperpolarization actually stimulates the closing of potassium channels, right? Because potassium channels are what's causing it to go back into repolarization, hyperpolarization. And that hyperpolarization is actually what shuts those. And then that also opens sodium gates immediately. So the sodium gates automatically open and we just start depolarizing again. Okay, so then we got this slow influx of sodium, which is leading to depolarization. Okay, that's going to go up until our action potential threshold is met at about -40 millivolts. Okay, and then that triggers the influx of calcium. Okay, so here's where it differs from everything so far. Right, so the slow influx of sodium right after repolarization till the threshold, and then that triggers opening of calcium channels. Calcium rushes in, causes the rest of the depolarization. Okay, but then luckily one thing's not different, which is repolarization. Again, it's still potassium exiting the cell like it is for all of them. Okay, so then we got one more action potential labeling on the next slide. If you're done writing this stuff down, feel free to hop over to that and fill that in, and you can answer that question there on your clickers as well. On the what?
Principle of the cells itself, like how are these different from what we learned?
These cells, these autorhythmic ones?
Yeah, like are they just
Yeah,
floating around?
they're just found in these little bundles. So there's some up here, there's some right here, and then there's some right here. This is the SA node, right? So there's just a bunch of special cells that kind of look like cardiac muscle cells, but they don't do any of the contraction part. Really what they do is just depolarize, and then they start the action, they start this electrical signal for us, and that's their role. Yeah.
They have different action potentials and everything else that they can report.
Yep.
It's like
Yeah.
a whole different system.
Whole different type of cell, different type of action potential. Yeah. Nope.
Yeah.
All right, so send in that answer on your clickers there. Tired? So you're sending in a number here. Where does calcium influx happen? It died? Okay.
So I won't get hooked on
Did you try just like spinning your batteries around? A lot of times that works. Take them out, put them back in. That work? It's magic. I do it with our remotes all the time. All right, so send in that number. Make sure to hit the send button. All right. So somebody asked me a good question walking around. It's probably important for everybody to understand. Just to orient yourself, these are different types of cells than the cardiac muscle cells, right? And they just respond differently. Their whole role is to start electrical activity in the heart and then to allow that to spread down. They're kind of like cardiac muscle cells, but they don't contract. Their role is to start the electrical signal, okay? And the way that they have ion flows is different than the other ones we've talked about so far. So they kind of have a separate type of action potential here. Okay, so what'd you guys say here? So about 3/4 of you said 2 would be the place that happens, right? So notice it never flattens out down here, right? So we don't have a resting membrane potential. It doesn't go back down and stay resting until it gets a stimulus. It doesn't need a stimulus. It's going to depolarize on its own, right? So when this comes back down, this is going to trigger the closing of potassium channels. It would happen right here. And then the opening of sodium channels, right? And that's number 1. So we get those sodium channels immediately open once we get down to about -60. And so then sodium starts leaking in slower than it did in the other ones, right? Because it's not going straight up, it's going much slower, okay? But then we get to this threshold point of -40. That triggers the opening of those calcium channels. And then we get calcium rushing in much more rapidly. So that shoots up in the depolarization direction, okay? And then finally, repolarization due to potassium exiting again. Okay, so definitely number 2 for where calcium influx occurs. Okay, questions on that? Go ahead, Mallory.
It doesn't contract, what is the COT?
What is the
It
calcium?
doesn't like contract, like it doesn't have active myosin, like I guess.
Yeah. So yeah, its role, the calcium's role is just to change the membrane potential in this one. Yeah. Mhm. Calcium is one of the primary signaling ions in all cells. So I'm sure it does other stuff in there too, but all you need to know for this class is that it changes the membrane potential. Yeah. Other questions on that? Okay. All right, let's go ahead and answer this question here. Yeah, and just take a guess. I haven't really told you the answer to this yet, so we'll just make this one quick so we can move on. All right, go ahead and send something in there. All right. Okay, so we have a bit of a normal distribution there in your answers, which I would show you if it wasn't so tricky to switch back over the computer, but kind of a lot of people choosing lots of different answers, what I'm saying. But about 40% of you are saying C, which is true. That's why. It's because the SA node just depolarizes faster than the AV node. So it's kind of whatever kicks off first is going to be the one to drive it. Okay. All right, let's talk briefly about what an EKG is. Okay, so to measure the electrical activity in the heart, we use something called an electrocardiogram. Okay, either can be called an ECG, which would be more accurate with our word for cardiac or cardio, right? But the technology was invented in Germany, and the cardiac or cardio in German is with a K, and that's why it's the EKG. Okay, and so what it's measuring is something called voltage, right? We talked a little bit about voltage last time. Voltage was the potential energy in electricity, right? It's the difference in charge from one location to another. And that's what all the little pads are measuring, is just the difference in electrical charge from one pad to the next. Okay. And so this is going to pick up on depolarization, repolarization, all of those kind of differences in charge. Okay. So very, very useful in detecting abnormal heart rhythms, heart attack risk, as well as a heart attack actually happening. Okay. And basically for this class, what I just want you to be able to recognize the main lettered waves there. Okay. So the first one we see, it's called the P wave. Okay. And the P wave is representing atrial depolarization. Okay. So that's that first thing that's kicking off. Atria are depolarizing. Okay. And then we move into the next part, which has 3 waves in it. And it's called the QRS complex. And that's representing when the ventricles depolarize. Okay, much larger tissue, much larger disruption of electrical activity there. And so we see much larger spikes. Okay, and then finally the T wave, that's when the ventricles are repolarizing. Okay, so that's the basics of what you need to know for EKG so far. We'll do a cardiac physiology lab later on in the semester where you'll work with these a little bit more once you're done with the cats. But that's it for now. Questions on those so far? Okay. All right, so let's talk about the cardiac cycle a little bit. We kind of just walked through this when we did this. When we did this at the very beginning of the start of class. These images are a little bit different. Okay. What I'd like to do, and I can come back and give you the order at the end. But what I'd like you guys to review is kind of these questions right here. Okay, so we've got just a few minutes left, but go ahead and see if you can kind of identify a letter where each of these things are happening. Okay, so you got 4 questions on the left, 3 questions on the right. You're just plugging in a letter with each of those. Don't worry about anything on the clickers. All right, guys, since we're running out of time, let's come back together. This would be a good way for you to study, but I'm just going to give you give you the ordering here, which would be C, A, F, E, D, then B. Okay, so that's just kind of the order. Those black arrows represent contraction. Okay, and then with this one, the big thing we talked about last time, the AV valves are closing when the ventricles contract, right? Because that sends the blood up underneath them, right? So when are the ventricles contracting to start with is in E, right? We see that right here. Ventricles contracting, that slaps these shut, okay? When the AV valves are opening would be right after the atria were kind of full, after ventricular contraction, when the ventricles are starting to relax, and we see the blood going from the atria to the ventricles, which would be C, right? Semilunar valves are going to close when the ventricles start relaxing as well, because we got blood pumping out of the heart, right, out of that aorta, and then the ventricles start relaxing. Blood's going to reverse when the pressure in the ventricles, it gets lower than the pressure in the aorta, right? Blood comes back. So where do we see that happening? Probably in B. All right, so we see the blood kind of hitting the back of those valves, trying to get back to the heart, but the valves stop it from doing that. Okay, that's a B. And then the semilunar valves open, not right when the ventricles start contracting, that's right there, but while the ventricles are contracting and we see blood exiting the heart, so that has to be D. All right, so we have to build some pressure before we get enough to actually open up the aortic valve. That's what's happening between E and D, is building up pressure so we can exceed the pressure on the other side of the aortic valve and open it up. Okay, so P wave would be which letter? Where do we see the atria contracting? F, right? P wave is atrial depolarization. QRS complex, we see both in E and D. And then T wave would be after ventricular contraction when we're done contracting the ventricles, and that would be sometime around B. Okay. This slide makes some noise, so it's kind of fun, but those are those hard sounds. We pretty much already answered this. When the AV valves close, that's our lub sound. We talked about that last time. When the semilunar valves close, that's our dub sound, right? So the lub is first, dub is second, okay? Because it's the blood hitting those valves. All right. Okay, guys, have a good lab tomorrow. Have a good rest of your day. See you Friday. Hey.
I was wondering if I could look at my test